Okay, this is Lecture 8, Neurophysiology Part 1. So we are going to introduce you to the basics of electrical communication in neurons today. This will be Part 1. So I'm going to give you an overview and reminders of how the nervous system is organized. We'll talk about electrical communication for Part 1, and then in Part 2 of neurophysiology in the next video, we'll talk about chemical communication at the synapses in neurons and the general idea of nervous system pathways. So the nervous system is the master controlling and communication system of the body. It is specialized to quickly detect and respond to stimuli. It's composed of the central nervous system, which is the brain and the spinal cord. And the central nervous system's job is integration, taking in information from the outside world, deciding what to do with it, and then sending out responses to the body so that the body can respond and maintain homeostasis. The peripheral nervous system is made up of peripheral nerves and receptors, and there are nerves that come into the central nervous system. Those are the afferent division. That is sensory information, so bringing in information. So touch information, temperature, pressure, pain, and then your special senses, hearing, vision, taste, smell, equilibrium, where our bodies are in space. All of that sensory information is coming into the central nervous system through the afferent division. Then, once the central nervous system decides what to do with it and integrates that sensory information and decides on a response, the other part of the peripheral nervous system will be the motor nerves sending signals out. And that will be signals out to the organs, out to the glands, and out to the skeletal muscle through the efferent division of the peripheral nervous system. The textbook organizes it this way. So we have the sensory stimuli coming in, bringing in information about what is going on through the afferent division. That will come into the central nervous system through the afferent peripheral nerves, where the brain and the spinal cord will decide what to do with that sensory information. And then if the brain and spinal cord decide to respond, then they will send out signals to the efferent division. And the efferent division we can divide up, whether it goes to the organs, that is the autonomic nervous system, or to the skeletal muscle. And the skeletal muscle will be somatic, or the body nervous system. That will go to motor neurons and then to skeletal muscles, and the autonomic will branch to a couple of different regions, the sympathetic, the parasympathetic, and the enteric nervous system. Enteric will go to the digestive system. Sympathetic and parasympathetic will go to the organs, where we will either have a stress response or a rest and digest response. Hopefully you guys remember all of that from anatomy. If you don't, please let me know. I can give you some links to review the anatomy of the nervous system. So overall, the functions of the nervous system are electrical and chemical communication. And as a part of that, we have sensory input, or bringing sensory information into the brain and spinal cord, integration within the brain and spinal cord, and then motor output, or signals telling the organs, glands, and skeletal muscles how to respond. So if we were to drill this down to the simplest of possible pathways, for example, this could be a very simple reflex pathway where we'd have only 2 or 3 neurons involved in the basic nervous system function. Then we could start with a sensory receptor, which will receive information. For example, this could be a touch receptor, which when it's pressed, it will send electrical signals through the afferent neurons. Those afferent neurons then will go into the spinal cord in this pathway. In the spinal cord then, we'll have the central nervous system interneuron neurons, which will integrate that information and decide whether they got enough electrical activity to respond. If they got enough electrical activity to respond, then they will send signals out to a motor neuron, which will contact either a muscle or a gland. And that could be smooth muscle, skeletal muscle, cardiac muscle, or glands. If they have enough information, they'll send signals out the efferent neurons, and that will activate the effector organs. So if we look overall at how the nervous system is organized, I want you guys to get very used to talking through these kinds of pathways, just in a general sense, because what we're going to do after we learn about electrical and chemical communication is we're then going to learn about some pathways in the spinal cord, some pathways in the brain, some general special sensory pathways, and we'll apply this basic organization to each of the parts of the nervous system that we study. So we'll start with peripheral receptors that are our sensory receptors. Those will signal and bring information, activating sensory axons, which will be contained in sensory nerves. Those will be afferent neurons, nerves going into the spinal cord and the brain. And then the spinal cord will either relay the sensory information to the brain, which is a typical pathway, or in the example of a very simple reflex pathway, the spinal cord can also act on its own and respond and simply send signals out to the muscles. Generally, though, the spinal cord will relay information to the brain. The brain will have some specialized regions to process and integrate sensory information and decide how and whether to respond to that sensory information. We will then send out motor commands back down through the spinal cord, or if the brain is acting on its own without the spinal cord, it could directly go through cranial nerves, and then to the muscles, organs, and glands through peripheral motor nerves. They will carry motor commands to the muscles, organs, and glands, which will be the effectors carrying out a motor response. I also want you guys to notice how similar this is to a homeostasis pathway. So remember back a few lectures where we talked about a stimulus, a integration, and response portions of homeostatic pathways. So the nervous system is generally carrying out homeostasis by activating, integrating, and responding to stimuli that are outside of the body. Okay, so we also need to review nervous system cells. There are two major cell categories in the nervous system: neurons and glia. Now, the technical term for glia is neuroglia, but most of us just refer to them as glia. So neurons are the primary signaling and communicating signals. We are going to spend most of our lectures talking about neurons, but I want you to know that there are also glia. And the research currently is looking more and more into the roles of glia, which are becoming super fascinating and much more than we previously thought glia could do. When I started working in a neuroscience lab years ago, we used to get our cells and we would plate them on dishes and we would throw the glia out and just keep the neurons and then just make sure that we had some glia maybe underneath to support the neurons with nutrients. Well, we now know that that maybe was not the best way to do things. Glia are doing much more than we previously thought. But for our purposes, we're going to focus on neurons here. And we'll tell you a little bit about the functions of the glia. Should have done more on this in your anatomy course, so review that if you need to. But the glia are the ependymal cells, the astrocytes, the Schwann cells, oligodendrocytes, and microglia. They are supportive cells that help with neuron development, growth, survival, and structural support. Whereas the neurons are going to be the signaling, both electrical and chemical signaling, and communicating cells. So here is just an overview, a reminder of these nervous system cells. You have the neurons, which are these large branching cells, and we're going to do the neuron anatomy again to remind you guys. And then surrounding the neurons, depending on where you are in the brain or spinal cord, you will have various glia nearby supporting. So here's an astrocyte, there's a microglia, oligodendrocyte, ependymal cells. Those are going to be supporting the nervous system, the neurons, in various ways. Okay, so neurons are the primary signaling and communicating cells in the nervous system. They are large branching cells that connect nervous system pathways both electrically and chemically. They come in many shapes and sizes, but in general, this is their structure. And let's draw out the structure of a neuron just to make sure you guys remember. Okay, so a neuron is a large branching cell, and the typical neuron is what we call a pyramidal or multipolar neuron, where we have the cell body containing the nucleus. So there's the nucleus, and there's the cell body. And then we have the input regions, which will receive signals from other cells. And these input branches are called dendrites. With several dendrites coming into the cell body, and they will bring signals into the cell. Those signals will get integrated here at the axon hillock. If they are enough, then we'll send large chemical signals called action potentials, which we're going to talk about today, down this single branch, which is an axon. The axon then, at its very end, will branch to the target cells, which will be other neurons. At the axon terminals. Where the axon terminal contacts dendrites or cell bodies of other cells, we call those connections synapses. Okay, so back to neuron structure. So the dendrites receive input from other cells or receptors. The cell body contains the nucleus. The axon hillock will integrate electrical input from the dendrites. It's a very highly excitable region. If we get enough electrical input, it will initiate an action potential, which will transmit down the axon to the axon terminal. The axon terminal will be the very ends of the axon, which will contact the next cell at a synapse, where it will release chemical neurotransmitters. Here's a textbook picture of a multipolar neuron with its many dendrites, with the arrows showing the input coming into the dendrites, and then the axon hillock here, which would integrate that information. And then if it's enough, we'll get the action potential traveling down the axon to the very end of the branches where we've got the axon terminals. That is then the output that will release neurotransmitter to influence other cells. So synapses are connections from neurons to other cells, and we have three basic types. We have a normal neuron-to-neuron connection that we just generally refer to as a synapse. If we have a neuron to skeletal muscle, we refer to that as a neuromuscular junction. And then we can have a neuron that can contact glands and cause release of hormones and other chemicals or secretions from glands, and that's a neuroglandular synapse. Okay, so today we're going to focus on electrical communication in this lecture. The next lecture will be chemical communication. But neurons can communicate both electrically through electrical gradients and chemically through neurotransmitters. Electrical communication happens through the movement of ions. Do you guys remember what ions are? Ions are charged molecules. So for example, a sodium ion has a +1 charge. A potassium ion has a +1 charge. A chloride ion has a -1 charge. So the movement of these charges in and out of cell membranes through the ions can be small, can be local, or it can be a long domino effect cascade of ion movement and electrical signals that can travel long distances down the branches of the cell. If it reaches the synapse will release neurotransmitter, which will then create changes in the next cell. That next cell could have changes either in contraction, if it's a skeletal muscle, release, if it's a gland, or more electrical signals, if it's a general synapse. So neurons, because they are electric, neurons are excitable. They're activated by stimuli to send signals to other cells. So a stimulus in the case of a neuron is any change in the environment that could trigger a change in the neuron. Typically, these will be sensed by different receptors, where we can have mechanical receptors, for example, touch, vibration, movement that will be detected. Electrical stimuli through electrical receptors, these are generally sensitive to ion changes or voltage changes. We'll get into how voltage is measured. Chemical changes, which are often used by taste or smell, or ligands from other cells. So for chemical changes, you may want to go back and review the cell communication lecture. Activation of neurons happens through activation of channels, proteins, so channel proteins that are on the dendrites. So a stimulus will come in, it'll change the shape of a channel. The channel will then transmit through electrical or chemical changes to make changes inside the cell. Very similar to what we talked about in the previous lecture for cell communication. We're going to get a little more specific than what we talked about in the previous lecture, though. So in the previous lecture, we talked about ion channels as a single group, where a chemical binds to a receptor. That receptor then makes a change, and if it's an ion channel, then that receptor will open up and let ions in. So that would be a ligand-gated channel. That was the example we gave in the previous lecture. But it turns out there are other ion channels that are found in neurons. There are some channels that don't need a ligand gate. They're kind of leaky. They're always open. Ions can move in and out of them, and they're specific to a particular ion. Mechanically gated channels will open and change electrical activity, let ions into the cell in response to physical changes. So a lot of these are, touch receptors or pressure receptors. Actually, hearing does this. So when it moves, that movement changes the shape of the ion channel, and the movement opens the ion channel. Voltage-gated channels are a little bit more difficult to picture, but they're very important in neurons. So voltage-gated channels are going to be channel proteins in a membrane that will change when the electrical activity around them changes. So you'll have ions and electrical activity changing nearby, and that electrical activity will cause a conformational or shape change in the channel that will open up the gate and let ions into that channel. So all of these are ion channels, and there's different ways to open the ion channels. For mechanically gated channels, we can push them open. For voltage gated channels, we change the electrical activity around them, and that causes the gate to open. For ligand gated channels, we have a chemical that comes in, and that will open the gate. And for leaky channels, the gate is just kind of always open, and the ions can move. So we're going to change electrical activity by opening and closing ion channels, and there's various ways to do that. Okay, so let's get really into the nitty-gritty of electrical communication. I'm going to warn you guys, you're going to need to probably go slowly through this one. Take some really good notes. We're going to draw a little bit too, but make sure that you really understand this before you move on. So electrical communication within a neuron occurs by the movement of ions. These are charged molecules like sodium and potassium that are moved in and out of the cell membrane. So ions carry electrical charge because they are charged, and there are different concentrations of ions at the cell membrane creating electrical gradients. So electrical gradients then are measured as something we call membrane potential. So membrane potential is defined as the amount of electrical charge that is present inside the cell compared to outside the cell. So let's draw this a little bit too. Okay, so for membrane potential, generally these experiments are done where you have an electrical wire or some other way, some electrical way to measure the inside of the cell. So we've got a cell that is sitting in a dish and an electrical recording apparatus inside. So it's going to measure the inside of the cell compared to what the cell is sitting in. Usually in these experiments the cell is sitting in a dish, or maybe it's sitting inside the brain or inside some glial tissue. So we're going to measure the inside compared to a reference electrode, which is outside. And this is by convention. We always care about what is inside the cell compared to outside. Why? Because a lot of times in these experiments, we've made the solution. We know what's in the solution. We care about what's happening inside the cell, not in the solution. So we're going to measure the electrical voltage inside the cell compared to outside. And that difference of in minus out is going to be your membrane potential. Just like the electrical activity in your house, we measure this in voltage, but these are really tiny cells. So rather than large volts, we're going to measure it in tiny volts or millivolts. So, by definition, a positive membrane potential means that the inside of the cell is more positive than the outside of the cell. And this can happen two ways. There could be more positive ions inside the cell than outside the cell. For example, you've got a lot of potassium inside the cell and not a lot of positive ions outside the cell. Or the flip of that is you could also have the outside of the cell have more negative ions. So think about that for a second, because this is in reference to the inside versus outside. So if the inside, say, is neutral, but you've got a lot of negative outside, then we would say that the inside is positive relative to that negative outside. Negative membrane potential is the opposite. This is when the inside of the cell is less positive than the outside of the cell. And there's two ways to do this. You could have more negative ions inside the cell, for example, a bunch of chloride, compared to outside the cell. Or you could have more positive ions, for example, sodium, outside the cell compared to inside. So again, think about this one for a second, and this is actually the case in neurons at rest. A negative membrane potential means you have fewer positive ions inside the cell compared to outside, or you've got a buildup of positive ions outside the cell. So membrane potential is measured in volts, but it's very small, so we use millivolts, which is 1/1000th of a volt. To calculate the membrane potential, we actually use something called the Nernst equation. The Nernst equation is not something you guys are going to need to use in this course to calculate membrane potential. If you go further and you have an interest in neuroscience, you will learn the Nernst equation. We're not going to do that today. The Nernst equation helps tell us the actual ion concentrations and how that relates to the charge. We're not going to do that here. Here we're just going to give you examples of differences in charge across the membrane. So the easiest way to do this is to look at the total amount of positive and negative charges inside the cell versus outside the cell. So here's a very simple example. If the membrane potential is 0 millivolts, that means that the inside and the outside have the exact same number of positive ions. They would add up to +10 on both sides, and the exact same number of negative ions, -10 on both sides. We would then sum them up and look at inside, subtract the outside, and we would see that 10 minus 10 is 0, -10 minus -10 is also 0. We have no difference of charge across the membrane. This will be neutral charge. More commonly, though, because we're talking about electrical signaling, we are going to have some difference. So here's an example of how we would get, say, a -10 millivolt difference inside a cell. So for that, we could have fewer positive ions inside the cell. So if the inside had only 5 positive ions compared to the outside having 15 positive ions, if we then subtract +5 minus +15, we would get a difference of -10. That is then assuming, of course, that the negative ions are equal and balanced. We only have a difference in this case of positive ions. So 5 minus 15 gives us -10. Again, inside minus outside gives us a difference of 10 millivolts across the membrane, where the inside was less positive, a value of +5, compared to the outside being more positive, a value of +15. I know I'm sorry to do math, you guys. We're going to have to do a little bit of math today, but you guys can do subtraction. Yeah, we can do that. Okay, so the inside is less positive, which makes overall the membrane potential -10. When this is the case, we do not have a neutral cell anymore. We will have charged ions accumulating at the membrane because of this new electrical gradient of 10 millivolts sitting at the membrane. Why are the ions sitting at the membrane? Think back to our membrane transport lecture. What kind of molecules can get across a lipid bilayer? Only nonpolar, small, lipid-soluble molecules can get across the membrane. So these are charged, in other words, they are polar. They cannot cross the membrane without channels. So these charged ions will accumulate at the membrane and create a potential difference or a potential movement of electrical activity if some channels were able to open. Okay, so here's a visual of those two examples we just gave you. So what you want to do when we give you these visuals, and it's probably a good idea for you guys to draw these out somehow that makes sense on your own. You're going to take all of the positive ions inside and subtract all of the positive ions outside. You'll take all of the negative ions inside and subtract all of the negative ions outside. In this one, we have 0 millivolts when you do that. All the positives balance, all the negatives balance. This membrane has neutral membrane potential. In this example, we have a 10 millivolt difference between the inside and the outside. So we take the positive ions that are inside the cell and we subtract the positive ions outside the cell. Then we would take the negative ions inside the cell and subtract the negative ions that are outside the cell. And our difference, really in this example, comes from the difference in our positive ions. So we took 5 and subtracted 15, and we got a difference of negative 10. So this example has a negative inside the cell. And I want you guys to notice that that negative inside the cell is not because of the negative ions. That negative inside the cell is because there are fewer positives inside the cell relative to the outside. Because those ions cannot cross, that charge will just build up at the membrane. So you'll have negatives accumulating on the negative inside, and you'll have positives accumulating on the positive outside. They can't cross, so this creates a separation of charges, which is our membrane potential, or our potential for electrical signaling if we get some channels to open up here across the lipid bilayer. So the ion accumulation at the membrane is what creates that membrane potential. When an electrical gradient, fewer positives in that example, is present at the membrane, the ions will accumulate close to the membrane due to the attraction of the negative to positive charges. So we really have an electrical gradient here now. So remember that ions, because they're polar, cannot pass through the lipid bilayer without the help of the channels, because the lipid bilayer has that large hydrophobic region that repels anything with charge, or repels anything that is polar. So the electrical energy is now technically being stored. It's building up at the membrane. It really wants to move. It's going to continue to build, but it can't move because there's no channel for it. Now it's going to be in the signaling that we have, opening up these ion channels, where we will get electrical movement in and out of the cell. So these electrical ions are waiting, storing energy, waiting for some signal to change. So we look at the overview of this, then if we have a negative membrane potential inside a cell, we have negative ions building up inside the membrane and positive ions building up outside the membrane. These separated charges will form a layer along the membrane that is just itching to equilibrate. It wants to change. Remember that cells want to be both chemically, due to chemical gradients, in chemical equilibrium. They want to be chemically neutral. They also want to be electrically neutral due to electrical gradients. So the increased number of ions that accumulate at the membrane potential will increase the magnitude of the membrane potential. So bigger differences, bigger membrane potential. Again, we would calculate the technical specifics of this using the Nernst equation, which you guys don't have to worry about. We're just going to give you some values, but we want you to understand where these values come from. Okay, so let's talk now, those were some general examples of membrane potential. Let's talk now about how neurons are set up so that they have a lot of membrane potential and a lot of potential to create electrical signals. So overall, neurons have a negative membrane potential. Are you shocked? That's why I gave you the negative example. I know, shocking. So they have a negative membrane potential at rest. And this is because of three main factors in neurons. There are a lot of negative proteins, which we're going to give an A- anions. We're going to just call them generally anions inside the neuron. There is a fair amount of potassium inside the neurons. We say a moderate level of potassium inside the membrane. But it's really this very, very high amount of positive ions, high sodium ions outside of neurons that give the membrane potential overall in a resting neuron generally a negative 70 millivolt membrane resting potential. Let's draw this out. Okay, so here is our membrane. We have the outside of the neuron compared to the inside of the neuron. We're going to have at rest in the membrane something called sodium-potassium pumps. Sodium-potassium pumps out 3 sodiums for every 2 potassiums in. At rest, these are all working all the time to ensure that this is the balance of sodium and potassium at the neuron's membrane. Lots of sodium outside, little bit of potassium inside. What that does is create a lot of positive ions outside the cell, a little bit of positive inside the cell. In addition to this difference in positive ion balance, we also have a ton of large negative anions inside the cell. So between the negative anions inside the cell and the high positives outside the cell, overall, when we calculate the difference between the inside and the outside of a neuron at rest, our membrane potential is going to be minus 70 millivolts. Now I want you guys to memorize that number and hold on to it. So this is the situation in a resting neuron. And these ions, the potassium ions and the sodium ions, generally are not able to change because we have sodium channels that are closed and potassium channels that are closed. So overall, neurons have a negative membrane potential of minus 70. So write this down, minus 70 millivolts in a resting neuron because we have high sodium outside, moderate potassium inside, and a lot of negative anions inside the cell. So taking into account the anions, the sodium and the potassium, a general neuron has a membrane potential of minus 70 millivolts. This is at rest when no electrical signal is occurring, and this is what we call the resting membrane potential. So we're going to set it down here, negative 70 millivolts at rest, and we're going to go up and down based on some signals. So all changes in membrane potential due to signaling are now compared to rest, and we have some terms for this. So the proteins generally cannot move in and out of the cell. They're too big, they're stuck. So we're mainly going to focus on the ion changes, sodium and potassium. Now, yes, there are and there can be other ions in cells and in neurons, but it is generally the sodium and the potassium that give the major changes. If you guys take this further into some neuroscience classes, you will learn more about other types of ion channels and ion changes. For us, we're going to focus on sodium and potassium. So if positive ions move into the cell, then the cell is going to go from negative towards positive. That going from negative, rest, to a more positive or close to zero, even past zero, we call that depolarizing the cell. Now, if we're at rest at -70 and positive ions leave the cell, or we get less positive inside the cell, less positive is going to make us go even more negative. Anything below rest we call hyperpolarization. So hyperpolarization is becoming even more negative from the resting value. So the sodium and potassium ions then are affected by electrical gradients and their individual concentration gradients. So if we want to know now when a cell gets a signal, what's going to happen? So we want to then look at the individual ions and see how these ions could change. So first let's look at potassium. So for both of these, we want to look at both the concentration gradient and the electrical gradient. For potassium, remember there is high potassium inside the cell at rest because of that sodium-potassium pump working. So then potassium wants to move out of the cell because it wants to go from high to low based on its concentration gradient. There is also an electrical gradient for potassium. Potassium has a +1 charge. It's positive. What is the membrane potential inside the cell? -70. So the inside of the cell is negative. Potassium is inside the cell and it is positive. So actually, there is a small force, this negative membrane potential, that wants to keep potassium inside the cell. Now, if we did the Nernst equation, you guys would see that overall we balance the concentration and the electrical gradients. The concentration gradient is much stronger for potassium, and potassium overall wants to move out of the cell. So if potassium were allowed to completely balance out both its concentration and electrical gradients and reach its equilibrium, the cell would be at -90 millivolts. That's pretty close to rest. So potassium is actually fairly close to equilibrium at the resting value of the membrane. Sodium also has concentration and electrical gradients. Sodium is very high outside of the cell and it wants to move into the cell because it's going from high to low. So high sodium outside, low sodium inside. Sodium wants to move into the cell. The electrical gradient for sodium then, sodium has a +1 charge. What's the inside of the cell? -70. Sodium then has an electrical gradient also to move into the cell. So both the concentration and the electrical gradient are driving sodium into the cell. So sodium really wants to get into the cell. If sodium can balance and reach its equilibrium, then the cell would be at +60 millivolts. So clearly, at rest, sodium is very far from being at equilibrium. So another way of saying this is that potassium kind of wants to move. It's not bad. It would like to go from -70 to -90. Sodium really wants to move. It's so far from its resting, it's so far from its equilibrium potential. It wants to go from -70 all the way to +60. There is a big force on sodium to move. But all of this doesn't matter if we don't have any channels for these ions to move. So nothing moves without the channels. So before we get into the channels, let's just review this. So your textbook has some diagrams for this, so you can look at both the concentration and the electrical gradients for each of the ions. So here it is for potassium. For concentration, here's the inside of the cell. We have a high amount of potassium inside of the cell. That from high to low is a concentration gradient, showing the arrow this way, that is driving potassium out of the cell. There is also an electrical gradient for potassium. because the inside of the cell is negative and potassium is positive, there's a small electrical gradient pushing potassium inside the cell, and that's why this arrow is going this way. So actually the forces on potassium are opposing each other, but when we do the overall calculation, it is the concentration gradient that is the strongest. If we were to open potassium channels, potassium would leave the cell. Now let's look at sodium. So for sodium, we have a high concentration gradient moving from high to low for sodium to move into the cell. So sodium is high outside and low inside the cell because of that sodium-potassium pump setting everything up. So sodium wants to move into the cell for concentration. Now I'm leaving this up here because this is your textbook, but notice that this is a typo in the textbook. Or I guess it could be showing, I'm not sure exactly why they've done it this way, but what this really should show is the resting membrane potential of the inside of the cell being negative. And the electrical gradient should be pulling sodium also into the cell. So why the textbook publishers decided to do it this way, I don't know. So on your diagram, redraw this, okay? Put negative inside the cell, positive outside the cell. Sodium wants to move. This arrow should be going this way. Sodium's electrical gradient wants to move sodium inside the cell. And we are not going to talk about other ions like chloride yet. Okay, so let's talk about the channels now, right? So we're waiting, right? Sodium, potassium. Potassium sitting inside, it wants to go out. Sodium sitting outside, it really, really, really wants to come in, and we're stuck, right? We're stuck at this minus 70 millivolt membrane potential unless we get some channels to open. So let's look at what the channels look like at rest. At rest, we have both potassium channels and sodium channels. Turns out that at rest, we have some leaky potassium channels. So when the neuron is at rest, we have some passive, they don't require signals, potassium channels that are kind of always allowing potassium to leak out. This is actually why potassium is close to its equilibrium potential, or why one of the reasons why the inside of the cell is very negative. So this also means that the permeability, or the ability for potassium to move in and out of the membrane at rest, is high, but it's not complete. Potassium is not at its equilibrium. It would still move if we opened more potassium channels. Sodium channels at rest are closed. So there are very few open sodium channels at rest. So another way of saying that is the sodium permeability at rest is low. It cannot move because the sodium channels are closed. It is also very far from its equilibrium because of that. Okay, so overall, let's look at the forces that are driving ion movement inside the cell. And now this is much better. This is a better diagram from the previous sodium one. So overall, if we open potassium channels, potassium wants to leave the cell. It has an overall driving force to go out of the cell. Overall, for sodium, sodium wants to get in the cell. So overall, it has a driving force, both electrical and concentration, to move in. And as we look at the way that these are set up, we have a high amount of sodium outside, moderate amount of potassium inside, ton of negative anions inside. That gives us everything set up at rest at minus 70. Now that we know how things are set up at rest, let's take a look at how it is used to actively signal within the nervous system. So electrical signaling in neurons, again, takes place by changing membrane potential away from rest. This is done by opening or closing specific channels to allow ions, sodium and potassium, to move in or out of the cell. And that's done through different trigger or stimuli events. This causes a change in the cell, for example, neurotransmitter release, that can be read by other cells. Okay, so let's make sure you guys feel good about these electrical signaling terms. The membrane potential is the difference in electrical charge across the membrane. We're going to subtract the outside from the inside, or in other words, we're going to do the inside of the cell relative to the outside, and we will measure that in millivolts, or 1/1000th of an electrical volt. If there is separation of charge across the membrane, that's called polarization. The membrane is polarized. For example, -70 millivolts at rest in neurons. If a membrane becomes less polarized or goes closer to zero or more positive, it becomes depolarization. This is usually when positive ions move inside the cell. If a membrane becomes more polarized or further negative from its resting value, then this is called hyperpolarization. This usually happens when positive ions move out of the neurons. Repolarization is also a term that is used, and that is during signaling. As a membrane potential goes away from its resting value, repolarization is going back towards a resting value. So membrane potential at rest is this kind of situation. Lots of positive ions accumulating outside, negative accumulating inside. The intracellular fluid, or the inside of the cell, is more negative than the outside. And this is solely due to more sodium compared to potassium and negative anions inside the cell. Now we can have a couple of ways to change membrane potential. These are just small changes in membrane potential. So if we start at -70 and we go up away from rest, then we call that depolarization. Then we repolarize back down to rest. If we go further down away from rest, that's hyperpolarization. And then back to resting potential. So there's two ways that membrane potential can change in neurons. Small changes like those that I just showed you on that previous graph are called graded potentials. These are small local changes in membrane potential that can vary in size, usually 1 to 5 millivolts, and they stay very locally at the membrane. They're caused by small changes in nearby connected cells or receptors. They are used for very fine-tuned, intricate, small information, small inputs to the cell. But they can build up to become a larger trigger. If we get enough graded potentials in a cell, then we can build up to a large, fast, long-distance electrical signal that is called an action potential. This is an all or nothing signal that is a chain reaction of events that travels all the way down the axon to the axon terminal and is the same magnitude every time it occurs. This is caused by large changes and or multiple connecting cells or receptors changing, building up a lot of graded potential to trigger the cell. Once the cell is triggered, then this is the all or nothing signal that will communicate between cells. So again, graded potentials, small local membrane potentials due to some kind of triggering event. Often these are small gated ion channels that are open for a short time in a small section of the membrane. The ions specific to that channel will move passively with their gradients. These are going to be local current flows, changing the membrane potential in small areas, but the signals will dissipate with time and distance. They can be, if they're depolarizing or positive ions move into the cell, we call those excitatory. Often that is sodium moving into the cell. They can also be inhibitory. So this can either be negative ions move into the cell, for example, chloride, or positive ions moving out of the cell, for example, potassium leaving the cell. That would cause the membrane to hyperpolarize, and those would be inhibitory graded potentials. Whereas the excitatory potentials would be depolarizing potentials, or excitatory. So here's that picture again. These are small graded potentials where, for example, here we have positive ions moving into a cell through a small channel. That is going to be a small excitatory graded potential. Graded potentials will only activate small areas of membrane. They're not going to travel all the way down. So you'll see them change just local areas, but you won't see them triggering a domino of events like we will see when we do the action potentials. Okay, now the action potential is the important signal. This is a large, fast, long-distance spread of membrane potential change. They will build up from graded potentials. So, okay, the graded potentials, they're important, but they are small signals, small local signals. Graded potentials will build up until they trigger an action potential in the cell. So I like to think of graded potentials as like the annoying little brother, right? That's tap, 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 tapping. So the graded potentials are the little taps. Okay? If the cell gets irritated or annoyed enough by the graded potentials, then we get this explosion, right? You get super angry when you get tap, 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 like really angry, which is the action potential. So the graded potentials are going to be the tiny little taps that will activate a cell. But if they build up enough, then we'll get this big explosion of electrical activity, all or nothing, traveling all the way down the cell. That's the action potential. It will start at one triggered site and it will propagate down the entire cell without dissipating. It's going to look and be exactly the same every time it occurs, so it is a reliable signal. It's the same pattern, and it is one way down the axon towards the axon terminal or towards the synapse. Okay, now we're going to go through the phases of the action potential. We're going to talk about what happens as the action potential changes, what happens in the ions, what happens in the channels, and what happens to the value of membrane potential in these different phases. At the end, we'll put it all together and graph it. So we'll start at rest, we'll hit a threshold trigger, then we'll depolarize, repolarize, hyperpolarize, and return to rest. After we explain everything, this graph is going to become very familiar to you guys. This is what an action potential looks like when we go through all of those phases: resting, depolarizing or going positive from the negative membrane potential, repolarizing going back down towards rest, hyperpolarizing going below rest, and then returning to its resting potential so it can signal again. To understand this, we need to add a little bit more about the voltage-gated sodium and potassium channels that are present in neurons. So action potential events are caused by the opening and closing of sodium and potassium channels that are sensitive to membrane potential. When channels are sensitive to electrical activity or membrane potential, we call this voltage-gated, because it is the change in the voltage around that channel protein that causes the change in the channel and the opening of the channel. So we have in neurons both voltage-gated sodium channels and voltage-gated potassium channels, and this is how they behave. So these are their properties. So voltage-gated sodium channels will be closed at rest, -70 millivolts, so the protein is shaped such that the gate is closed. When the cell reaches -50 to -55 millivolts, then the sodium channels open. The voltage-gated sodium channels open. because the voltage changes the shape of the channel, opens up the channel, and sodium can now rush through that channel. In addition to the gate, sodium channels also have a plug, or an inactivation gate. Voltage-gated potassium channels will be closed at negative 70 millivolts at rest, and they have a gate that will open at minus 50 to minus 55. But the potassium gate takes a long time to open. It's kind of like when you drive into a neighborhood and you have these slow gates, right? You sit in front of the gate and it goes, right? The sodium channels open right away. Potassium channels, they take a little more time. So sodium will signal first before potassium, and they both have the same threshold, minus 50 to minus 55. The potassium channels do not have an inactivation gate or a plug. They only have this regular voltage gate that will, after it opens, it will then close slowly. Here are some visuals from your textbook of the sodium and potassium channels. So here are the sodium channels with their inactivation gate. So this is the voltage gate. This is the inactivation gate. So at minus 50 to minus 55 values of membrane potential, the sodium gate opens. As that then has time for the sodium to move, slowly this plug, or the inactivation gate, will start closing the sodium channels. Now I want you to be careful with the language here, because when we say closed, we usually mean the gate. When we say inactivate, we mean the plug or the inactivation gate. So the gate is open, but sodium cannot move through at this stage because it's inactivated. Eventually then, the inactivation gate will release and the regular gate will close. For voltage-gated potassium channels, very simple, just one gate. The gate is either closed or open. Okay, now let's go through the events. So first, at rest, the membrane is going to be at minus 70. That's because of what we have already set up, the sodium-potassium pump, the sodium gradients, potassium gradients, and the negative anions. And let's now follow the channels. Okay, so back to a membrane where we have the outside compared to the inside. And we've talked about a couple of different channels. We have some sodium channels, sodium-potassium pump that are going to let pump sodium out of the cell, pump potassium into the cell at rest. We've got our sodium voltage-gated channels that have a gate and an inactivation gate. So these are our sodium channels, voltage-gated sodium. Then we have voltage-gated potassium channels that just have a gate. And we also have some leaky potassium channels that are always there. We're going to kind of ignore these for the rest. Just know that potassium channels, there are some leaky potassium channels. Okay, so at rest, the membrane potential is minus 70 millivolts. So at rest, we have this situation. A lot of positive sodium ions outside the cell, a few positive potassium ions inside the cell. That overall gives us a resting value of negative 70 millivolts. So the potassium leaky channels are open at rest. The potassium voltage gated channels are closed at rest. The sodium voltage gated channels are closed at rest. And the sodium potassium pump is actively ensuring that we have this negative 70 millivolt membrane potential. Then we're going to get some input to the cell. So either through receptor potentials, through chemical signals, we're going to get some positive ions into the cell. If we get enough positive ions into the cell, we're going to call those graded potentials. And they will be taking us to what we call threshold. Threshold is going to be negative 55 millivolts. We call this threshold because at negative 55, the sodium and potassium voltage gated channels will be triggered open. So we reach threshold due to a buildup of graded potentials. Once we hit threshold, we're going to open up sodium channels. Which way does the sodium want to move? Sodium wants to move in the cell. So when we open up sodium channels, does the membrane potential get more or less positive? Remember that we're reading membrane potential inside the cell. So if sodium is moving inside the cell, we're getting a ton of positive ions into the cell. And remember that sodium was really far from its equilibrium, so there's a very strong force of sodium inside the cell. This we call depolarization. So it's going to depolarize, and it's going to go actually all the way up to around positive 30, positive 35 millivolts. Tons of positive sodium ions are going to move into the cell. It's going to stop and be limited at positive 35 millivolts when the sodium channels inactivate. So the rising phase. Sodium rushes in, potassium leaky channels always open, sodium voltage gated channels open. That's the important piece. Potassium voltage gated channels are closed because they've hit threshold, but they're too slow. They haven't opened enough yet for potassium to move. So the overall event here in this rising or depolarization phase is that sodium is rushing in. Now, sodium is going to hit its peak at positive 35. It stops at positive 35 instead of sodium's equilibrium potential of positive 60 because of this plug. So, as the voltage gate opens, the plug starts to tick, 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 and eventually plugs up and stops the sodium from moving in. At the same time, the slow potassium channels have finally opened. What happens when potassium channels open? Which way does potassium want to go? Potassium wants to leave the cell. When potassium leaves the cell, it carries positive charge with it. So this phase is a falling phase from positive back down to negative. And so this is a repolarizing of the membrane back toward minus 70 as positive ions now leave the cell. And that's potassium that is carrying that positive charge. So in the repolarization phase, we're going to move back towards -70 because of potassium moving out. Sodium voltage-gated channels will be inactive, not closed, inactivated. Potassium voltage-gated channels will be open now. Now, potassium voltage-gated channels don't inactivate, and they close very slowly, so they are going to continue to hyperpolarize, going even further to -90 millivolts. So potassium is going to continue to leave the cell beyond the resting value, past -70 to -90. At that point, we call this the hyperpolarization phase, and it's all due to potassium moving out of the cell. Then the final resting, or the final return to equilibrium, is going to happen as the potassium channels then close. The sodium channels will fully close, and the sodium-potassium pump is going to reset everything back to rest at -70. So let's look at this then in a graph form. So remember our key values that we just talked about. Membrane potential at rest is -70. Sodium rushes in until about +30, +35. Then potassium leaves the cell to bring it back down, repolarizing to -70. Potassium then continues to leave the cell down to -90. So at rest, the voltage-gated channels are closed, and we're going to be at -70. We're then going to have a depolarizing event, which will be graded potentials adding up to threshold. At threshold, the voltage-gated sodium channels will open. Sodium rushes in, causing that depolarization from -70 up to about +30, +35. Then the sodium channels are going to plug up or inactivate, and sodium will stop moving into the cell. At the same time, the potassium channels, kind of late to the party, will finally open, and potassium will start leaving the cell. As potassium leaves the cell, potassium out creates the repolarization or the falling phase in this graph, which is going back towards -70 towards rest. Potassium is slow to close, so the further closing, the further movement of potassium out of the cell as the potassium channels are still opening is going to be this last hyperpolarization phase. At this point, the sodium channels are reset, the plug is reset, the gates are closed. All we need to do now is close the potassium channels. When we close the potassium channels, the sodium-potassium pump takes back over, and we get back to rest. I want you guys to take a moment to draw your own graph. Draw millivolts on your y-axis, time on your x-axis, and make sure that you understand where these numbers are coming from. So pause this and take a few moments to draw out your own action potential. Okay, the last bit before we're done is that there are some points during the action potential that are limits to electrical signaling across cells. So remember that an action potential is a traveling electrical signal, and its goal is to get neurotransmitter release at the synaptic terminal. There are 2 times in the action potential where no other action potentials can occur during this time. So an absolute refractory period is when the sodium channels are either maximally open or inactive. So look at your graph, and when are the sodium channels maximally open or inactive? That is during the rising phase, which peaks, and then the repolarization phase when they're inactive. There's also a relative refractory period. A relative refractory period is not absolutely no action potentials. It just means it's very, very difficult to get another action potential. So this will only get an action potential, a second one, if the triggering stimulus is very large, because only a few sodium channels are available and potassium is exiting the cell. So look on the graph. When is potassium leaving the cell and sodium channels are starting to reset? This is actually during the after hyperpolarization, or what we call the dip phase. At this point, you could have a second signal to a neuron, but it needs to be a very strong signal to get two signals close together. So if we highlight those areas, once you start an action potential, you cannot start another one during the absolute refractory period. Once the action potential is starting to hyperpolarize, you can get a second action potential, or another signal can activate the neuron, but it has to be much stronger, and that's the relative refractory period. This is important for understanding how different stimuli can, timing-wise, affect the cell. So as a review, graded potentials are input to the dendrites. The dendrites will receive multiple inputs, causing graded potentials to build up and excite the cell. If enough excitatory input is received, a threshold will be reached at -55 at the axon hillock, and the action potential will then travel down the axon to the axon terminal. So overall, we had some graded potentials in our dendrites. They caused a trigger or threshold at the axon hillock that caused the chain of events that we call the action potential, which will ultimately lead to signaling at the axon terminals through the synapse. So the action potentials then are going to spread all or none once they're triggered at the axon hillock. And once a group of sodium voltage-gated channels reaches threshold, the sodium will rush in, bringing huge patches of membrane to threshold, creating this domino effect of opening more and more voltage-gated channels down the cell, causing this domino of action potential. Action potentials will only move in one direction, down the axon towards the axon terminal. Part of that is because of the inactivation of the voltage-gated channels. In other words, once we tip the dominoes, they continue to tip down the axon. And it takes time before the dominoes will reset, which makes sure that the action potential propagates in one direction down the axon. Your book diagrams this. I'll tell you guys, I don't love these diagrams, but ultimately what you can see is that the graded potential gets you to threshold. Once you get enough, then you'll get this domino, which will travel, sending in sodium as the sodium channels open in a domino effect down the axon, ultimately reaching the axon terminal. Last two factors influencing action potential conduction is myelination, which you guys can think of as insulation on the wire, and the size of the axon. So myelin is a material that is a thick, lipid-rich layer formed by glial cells called oligodendrocytes, which are in the central nervous system, or Schwann cells, which are in the peripheral nervous system. They insulate the axons, and there are no ion channels underneath the myelin. So there are chunks of myelin with spaces in between. Those spaces have the collections of the voltage-gated sodium channels, and so this domino effect is going to jump from node to node down the axon. We call that saltatory conduction because the ions and the electrical charge only move at these nodes. And the voltage-gated sodium and potassium channels will be concentrated at these nodes, increasing the efficiency, jumping from node to node. And the insulation of the myelin ensures that this saltatory conduction is as fast and efficient as possible. This is a picture of myelin created by a Schwann cell. So here you have the nucleus of the Schwann cell, and then the myelin is wrapped around the axon. The last piece is axon diameter. So the bigger a cell, the faster the conduction, because there's less resistance. So big insulated neurons will be super fast, and you can get up to 120 meters per second. For example, in some motor neurons to skeletal muscle, that's 268 miles per hour that these action potentials can travel. Smaller axons, especially if they're unmyelinated, will be high resistance and they will conduct slower, and they will go 2 miles an hour or 0.7 meters per second. These will be slower signaling, for example, neurons to digestive organs. So space is limited. If an axon needs to be super fast, it's going to be both large diameter and myelinated. That's it for today. I think that's enough. Lots of information. Please let me know if you guys have any questions.